Thanks a lot, Dominica, and thanks for the invitation. So for those who do not know me, I'm head of the Bioinformatics Core Facility at the University of Fribourg here in Switzerland, and also group leader of the Swiss Institute of Bioinformatics. Can you see my pointer? Yes, I guess. Yeah.
Yeah.
Okay, great. So today I'm going to show you a little bit of what I do as part of my duty. So helping researcher to analyze their data. And I call it the grand tour or small tour of methylation, because in Switzerland we have a very famous, we have many of these picture frames that you can find in all the touristic places around Switzerland, that you can always take beautiful pictures from these places. So it's kind of a game to collect all the different frames, and I don't know how many hundreds of these we have in Switzerland. Okay, so for this grand tour of DNA methylation, today I will concentrate mainly on the prokaryotic methylations, because I don't have too much time to speak about the rest. So to remind you about DNA methylation, there are three types of, or most important types of DNA methylations that are found here, 5-MC, mainly in eukaryotes at the CPG islands. We have also 4-MC, which is occasional in prokaryote, and the most important in prokaryote is 6-MA, which is very frequent in prokaryote and a bit rare in eukaryotes, where it's found only in mRNA. The role of this DNA methylation is very variable, but I would say that the common theme is gene regulation that is found in eukaryote and prokaryotes, and also defense against phages in prokaryote, and also limit of transposon replication, which we can also see as a defense mechanism in eukaryotes. There are different methods to look at DNA methylations. One of the methods is called methylation sensitive restriction enzymes. So restriction enzyme that can, or that will cut only if the DNA is not methylated, or if the DNA is methylated. I mean, there are two options. You can, and with these methods, you see with this yellow color here, is that it's limited to certain motifs that can be identified, and these are of course dependent on the restriction enzyme that cuts these motifs. You can also use immunoprecipitation to detect methylation, and this works for the two main methylation types. And of course, the bisulfite sequencing is the most important method for eukaryote DNA methylation detection. Then with long-read sequencing, it's known now that PacBio and also Oxford Nanopore can detect directly DNA methylated bases during the sequencing process. And this shows that PacBio is capable to detect very easily 4-MC and 6-MA, also 5-MC, but usually it requires a higher coverage of the region, whereas Nanopore is also limited by the motifs. So for Nanopore also, there are some motifs that are easier to detect than others. So let's have a look at the first example. And in this example, we use a restriction enzyme methylation cutting. And in this grand tour, so this is a recent publication with some colleagues. And in this publication, they were interested by vaccines against Salmonella typhimurium. And these vaccines often show that typhimurium can be selected for evolved clones. So yeah, I mean, the classical, it's like with corona. I mean, you have variants that can escape the vaccine in that case for bacteria. And these variants were sequenced, and so for those, for some variants, mutation could explain the leak. because some of the enzymes like OAFA, for example, was mutated, explaining the disappearance of or the modification of the external glycosylated membrane here. And some other variants could not be explained by any mutation. And so they were interested to look at potential methylation analysis to see if this could explain the leak of these clones escaping the vaccine. And for that, we used restriction enzyme with MB01 that cuts at GATC location only when the A is unmethylated. And once you have cut, you can then add your adapter sequences and build your Illumina library and then sequence the reads. You remap the read to the reference genome and you can identify regions that have been found as being unmethylated. And so this is what we did for several clones. And you look here at 2 positions, A and B's, and these positions were identified as being the most differentially methylated positions in those clones compared to the reference genome. And so when the A position is heavily methylated, like it is the case here for the avirulent clone here, it means that the operon GTR-ABC is silent. When the methylation switched from position A to position B, like it is the case for these clones here, evolved clones, you see that GTR-ABC is transcribed, and GTR-ABC has an impact on the immune response, and these clones can escape because they lose some modifications of their glycosylation outer membrane protection around the membrane. And so this explains easily with this, you can read more in details in this article, how in fact the bacteria can evolve both based on direct mutations, but also due to simple methylation, DNA methylation changes. Another example that we looked at is prokaryote methylation with PacBio, and here we looked at 3 possible methylations. And this is a collaboration with a group of Vet Swiss Institute, where we looked at Staphylococcus strains that were sequenced with PacBio. And these strains are coming from animals, mainly camels. And these camels have several strains that show different motifs. So we could identify a series of motifs, about 70 motifs, different motifs here that you see at the bottom. And these motifs are differentially methylated in the different clones. As you see here in dark blue, you have the region motifs that are 100% methylated in the genome, and in lighter blue, motifs that are less methylated, between 25 and 50% within the genome. Yes? Yes?
Lorena, I am sorry, but you have already...
oh, I didn't hear the bing.
Yeah, indeed, because Lubos deactivated it and given me the role of the bell.
Okay, sorry. So I just wanted to show that we can also identify some heavily methylated regions within the genome using our tool called Pacman. And with this, we were able to identify resistance genes and also virulence genes within these different strains. That's it, basically. So thanks a lot.